I welcome you all to the class of biology. So, dear students, please report your attendance and we'll begin right away. Students, Shillong Center, Tamenglong and Itanagar, please report your attendance and we are beginning the class right away. Students, please get settled quickly. Please get settled quickly. Okay, yeah, I'm waiting for you to settle down. Hello, Tamenglong students, how are you? Okay. All right, Tamenglong, thank you. Other centers, Shillong centers and other centers, please report your attendance. Okay, thank you, Itanagar. All right, only Shillong center is remaining for the attendance. Shillong students, please call the others. I think they are just waiting outside the class, I believe, because I can see so many bags out there. They haven't come in, I believe. Okay. So please settle down quickly. And now we are beginning the revision of what we have done in the last class. So in the last class, what I have told you that there are many cell organelles that I had discussed. Okay, I discussed about plastids, chloroplast, mitochondria, lysosome, Golgi apparatus, right? So all these things we had discussed in the last class. Okay, so we had discussed the GERL apparatus, the Golgi body endoplasmic reticulum lysosome system. So I told you about the nucleus and Golgi apparatus nucleus and endoplasmic reticulum network. So first is the rough endoplasmic reticulum, then is the smooth endoplasmic reticulum. Okay, smooth endoplasmic reticulum, it makes lipids. Rough endoplasmic reticulum makes the proteins. So we have discussed this. Golgi apparatus is the packing and processing center, alright. And finally we discussed lysosome, the suicide bags, the things that are filled with acidic hydrolysis enzymes, acidic hydrolase enzymes, alright. So all these enzymes can digest almost any substance. So they recycle various organelles like old mitochondria also. Then we discussed about mitochondria. They function in respiration. Mitochondria take an important part in the aerobic respiration. So we have discussed this in the last class. Okay. And after that we had discussed about the mitochondria having the semi-autonomous, semi-independent cell organelle nature because they have their own genetic material and the 70S ribosomes. After that we discussed plastids. Plastids found in all plant cells, all euglenoids. Okay. And there are three types: chlorochromo leucoplast. Chloroplast have chlorophyll and carotenoids. Chromoplast have only carotenoids. Leucoplasts do not have any pigments. So they are of many types: amyloplasts, eleoplasts, alluroplasts. Okay. All right. So about this, you can see here, right? Okay. Now after chloroplast, we have other things. I had discussed you about ribosomes, the protein manufacturing factory of the cell. So they have two subunits, I told you, and I told you their importance in the protein synthesis. This much we will never forget, right? So this is very important that they have something called S value. I told you sedimentation coefficient. Actually, if I try to physically find out the dimensions, you have done dimensional analysis in physics. So if I look at the formula, sedimentation coefficient has units of time per second, okay, per second or per time unit. So it basically expresses how much time a unit of a cell will take in coming down to the bottom, or sedimentation value traveling that distance to the bottom of the test tube. So that is what basically sedimentation coefficient is all about. So it is independent of you cannot apply mathematics rules like addition, subtraction to the ribosome units because it is a time unit. So it will settle with its own time. Okay. So these are the things. And then I told you that ribosomes manufacture protein. They have two subunits. Large subunit has two subunits, has two places where amino acids can get attached. I told you this in the last class. These amino acids form peptide bond, then amino acid moves further, it reads a new messenger RNA information, picks a new amino acid, forms second peptide bond, third peptide bond, and the peptide bond chain keeps forming. That is why proteins are called polypeptides. After this, now today, what you have to do is cytoskeleton. Today we will continue with cytoskeleton. Okay, cytoskeleton. We will finish this chapter shortly. So let's start with cytoskeleton now. Students, do you want these notes? Yeah, I can also give these notes to you that I am writing down here in the class. Okay, so if you want them, I'll give them to you. Alright, so let's take a look at cytoskeleton. Alright. So please take a look here. In this picture, you can see there is a mitochondria here, right? Yeah, I'll give these notes to you, okay? I'll post them in your groups. We have some microfilaments, microtubules and intermediate filaments, and this unit structure, okay, the microfilaments, okay, and the sheet-like, tile-like structures, it is the cytoskeleton. I told you that cytoskeleton is the thing which holds all the cell organelles in its proper position. If cytoskeleton was not present, then nucleus, all the cell organelles, they would have come down bottom, crashing down on the cell membrane. It is because of cytoskeleton that cell has a specific shape. Otherwise, it would be just like a polythene bag and you drop it on surface of floor, then the polythene bag will flatten out, right? But if the polythene bag is, you have tightly sealed the polythene bag and then drop it, then it will have some shape. Otherwise, it will the water will all come out. Okay. So if the polythene bag is open and you drop it on the floor, all the water will come out. So it will become flat. That would have happened to the cell if cytoskeleton was not present. But because cytoskeleton is present, so cell will have a definite shape and each organelle will have its specific position maintained by the help of this cytoskeleton. Okay. All right. So cytoskeleton is similar to what we have in our okay. Cytoskeleton is similar to the framework we have in our body called the endoskeleton. So just like we have bones in our body, cytoskeleton is the basic bones of the cell. Okay, that is why it's able to maintain its specific shape. All right, so we have some information here. Let's write this information and the other information that I'm going to give you now. All right, cytoskeleton. So please write about cytoskeleton. It is an elaborate network. It is an elaborate network of filamentous protein filaments consisting of microtubules, microfilaments, and intermediate filaments, present in the cytoplasm. cytoplasm. Okay. All right, Shillong centers, please, I believe, yeah, more people are coming in from the outside. Now you can, I believe, report your final attendance, right? I can see there are about more than 13, 14 students, okay? So you can count and let me know. All right. Because I believe you people have your late lunch, okay? So what is the function of cytoskeleton? Function of cytoskeleton. Okay, those who have just come now, please complete this information. I have just begun writing these things down. Up till now, I was just giving the revision. So please write. The functions of cytoskeleton are: the cytoskeleton helps in mechanical support, helps in motility, movement of the cell is assisted by cytoskeleton, okay, like amoeba, all right. Motility, support in mechanical form, so you can write mechanical support. This is the most important function of cytoskeleton. Mechanical support, the most important function. The mechanical support, okay, and maintaining cell shape. Maintenance of cell shape, etc. Okay. So this is the function of cytoskeleton. So I just showed you that what cytoskeleton looks like. Okay. So this is what cytoskeleton looks like. Right. So there are many microtubules, microfilaments, so many things like that. Okay. So this is what cytoskeleton looks like. All right. And regarding the notes, I have already given you. Right. So this is about it. All right. So let's. Yeah. Okay. Thank you, Shillong. All right. Now let's put the heading of cilia and flagella now. Cilia and flagella. Cilia and flagella. Second last cell organelle. After this we cover nucleus and chromosomes, and after that this chapter will be over. Cilia and flagella. So what according to you is the function of cilia and flagella? What according to you is the function of cilia and flagella? Cilia has two functions and flagella has just one function. Okay, cilia has one function, sorry, two functions, and flagella has just one function. So what is it? Not just movement. Cilia and flagella both have common function of movement, but cilia performs one more function. What is that? Cilia performs one more function. What is that? No idea? No, that's totally wrong, Itanagar. I'll explain. Look, there is a huge confusion going on. Most students have this confusion whenever I ask this question. I'll clear this concept right away. People get confused between cilia and villi. People get confused between cilia and villi. Okay, so I'll explain to you what the difference is. Villi what is villi and what is cilia? Cilia is a structure formed on the surface of the cell, like there are ciliated epithelial cells, cilia. Now cilia has a tendency to move. Cilia has a tendency to move the surrounding fluid around it. Okay. Cilia are hair-like structures. Cilia and flagella both are hair-like structures, and this has a movement property. It can move in this direction, then it folds itself, and then comes back, and then performs this movement again. So I can say, first it moves in particular direction, then it coils, turns back, and then performs the movement in this direction, this way. Okay. It does not go back like this, because if this movement happens, no movement will happen. And what is this movement we are talking about? We are talking about the movement of fluids around the cell. Cell is having some fluids around it, and with the help of cilia, it can move the things around itself. Flagella and cilia both perform movement of the cell, or cilia can also control the movement of water or any other fluid surrounding that cell. Yes. Yes, Shillong students, you got it right now. Okay. Cilia help in your nasal passage. Some mucus is already forming. Okay. Yeah, most students have the confusion between cilia and flagella. It's perfectly fine. I'm here to clear your doubts. Okay, don't worry about it. So the thing is, cilia helps in the movement of mucus continuously in upward direction in your nasal passage. Okay, so in your nasal passage, whatever the mucus is there, we do not want that mucus to enter the lungs. Why do we produce that mucus? To prevent bacteria, viruses, foreign particles, dust particles, carbon particles, in general pollutants and pathogens, and this pollutants and pathogens, this will stick to the sticky mucus, and this mucus is eliminated from time to time from our nose. And you know what this material looks like, right? Nobody wants to talk about it. Okay, so it has a very dirty appearance because it's having a lot of bacteria, a lot of viruses also maybe, and a lot of dust particles, pollutant particles. So it's not a good thing, and we normally eliminate it when nobody is looking, right? So this is due to the movement of cilia. If the movement of cilia is stopping, then the problem can happen that this material can go downwards. So that is why cilia are continuously beating upwards. Beating upwards, then coiling. They do not want this material to go down. They come down, then move upwards. And this type of movement is performed on all the 360-degree round surface of the nasal passage or the bronchial passage. Another thing, in the females, in case of fallopian tubes, the egg is forming and the egg is coming, and the egg is traveling inside, finally going to the uterus. So what is helping the movement of the egg cell? The egg cell is supported by the movement of, by the touch of cilia around that egg. Cilia direct the egg to go from this place to the other place. Alright, I hope you have understood now. So cilia has many functions, not just preventing foreign matter. It has, for example, it plays a role in reproduction, in controlling the movement of the egg cell. Okay. So these are the things that you should be knowing. And on the other hand, what is villi? Villi is a projection. Villi is this like thing. Villi is finer than cilia. And it does not have a movement function. It does not have a rotating function or anything like that. It has only one function, which is to increase the surface area. It will lie perfectly still. It does not move like this. It lies like this only, and whatever food is coming from the outside or whatever substance is coming from the outside, villi has this property of increasing surface area. So due to increased surface area, more substance will be absorbed, right? For example, if you have a old towel and a new towel, so new towel has lesser surface area, it has more starch, it has lesser surface area. An old towel would be the best for drying your hair, drying your body. You know this very well, right? Because old clothes have more fibers, more bristles, and they're more spread out. So they have, you can compare that to villi. A new cloth has very less fibers, very less fine fibers. Old cloths have lots of fine fibers. You have seen them, right? Everybody has seen them, I believe. So if you're using an old cloth to wipe your face, it will do it faster because it will absorb the water faster from your body. But if you're using a new towel, you know how that feels like, right? Okay. So that was what I was trying to tell you about. So cilia and villi, now the confusion is finished. Cilia has another function. Villi has only one function, which is of absorption. Cilia plays a part in movement and controlling the rate of flow of particles around it, of materials around it. Movement, controlling the flow of movement or flow of fluid around cell. Fluids present around that cell. Okay? So this is the difference. So please don't get confused from now on. It's really easy. There's nothing special about it. So now let's continue with cilia and flagella. Cilia and flagella. So please write, cilia and flagella are hair-like outgrowths. Cilia and flagella are hair-like outgrowths of the cell membrane. Are hair-like outgrowths of the cell membrane. Cilia are small structures which work like, look, everybody has seen a race, a boat race in which lots of people sit on a large boat in South India, and there are lots of festivals like that, but there is a festival called Onam, and on that festival lots of people sit in 2 rows on a large boat, and they use the oars to move the boat faster. So there is a boat race festival in a festival called Onam in Kerala, and in this you can see lots of people are moving oars together. That is the situation similar to how cilia are operating. Cilia operate in this similar fashion. They move the cell in the coordinated way. So because if all the cilia are moving in, every cilia is moving in different direction, the cell will not go anywhere. Okay, the cell won't be able to go anywhere. But if they are moving in a coordinated manner, then all the cilia will propel the cell forward like paramecium. Okay, so which work like oars, causing the movement of either the cell or the surrounding fluid. Or the surrounding fluid. Flagella are comparatively longer. Flagella are comparatively longer and responsible for cell movement only. Responsible for cell movement only. So while I was teaching you bacteria, I had shown you what flagella are looking like, right? So flagella are long thread-like structures. Okay? Now let's take a look at the internal structure of cilia and flagella. Okay? So I have written this, right? Everybody has written it. So now let me draw a cross-sectional diagram of cilia and flagella. 1, 2, 3, 4, 5, 6, 7, 8, 9. Just a second. Okay. So this is done now. These are two interlinked together. This is how they are attached. All right. One, two, three, four, 5. 1, 2, 3, 4, 5. 1, 2, 3. Sorry, one more thing. Because of this circle, it's a bit difficult to observe, so I've erased the circle from here. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So all these spokes are forming you can join the circle. Just wanted to show you something. That is, I had drawn this. So in this book of yours, NCERT, the diagram is given. So there is no particular problem about it. Now let's start the labeling. This is the central sheath. Central sheath. This is the radial spoke. Radial spoke. This is the interdoublet bridge. Interdoublet bridge. And one more thing. This is the plasma membrane. Cilia and flagella both are surrounded by plasma membrane. and this is the central microtubule. Central microtubule. If you are having a difficulty in drawing it, let me make things a bit more clearer. The central doublet bridge is meant for connecting them together. So in the book your diagram is given, so no need to get worried about it. Okay, so I will just show you. Now let me show you how it actually looks like. Okay, so let me show you how it actually looks like. Okay. So this is how it has this structure. It actually looks like. So on the outside there is the plasma membrane, and now you can see very clearly there are 9, 1, 2, 3, 4, 5, 6, 7, 8, 9 peripheral double protein structures. Now these protein structures run parallel, so it is basically like a hollow rod running through the whole structure of cilia and flagella. These are not small rings. These are not small rings. These are hollow rods. So if I cut a section of a hollow rod, a double hollow rod, it will look like this. Alright. And these are 9 hollow rods of 9 double hollow rods and one central hollow rod having double microtubules. This is called 9+2 arrangement of the cilia and flagella. Okay. 9+2 arrangement of cilia and flagella. So you can see in this figure, right? And on the outer surface, the plasma membrane is present. Now, where in the, you can see this picture now. Now, please try to answer me. Where can you find cilia and flagella in your own body? Where can you find cilia and flagella in human body? No answer. Yeah, please try to answer. I have shown you the hints that you need to know. Nasal passage, stomach. No, you're mistaken about it. Stomach does not have cilia. Stomach does not have ciliated cells. Nasal passage has it, but not in stomach. Stomach has a different mechanism. Stomach does not need cilia. Okay. Fallopian tube, cilia. Nasal passage, cilia. But where can you find flagella? Where can you find flagella in your body? In which cell? Do you have any cell which has flagella, yes or no? Do you have any cell which has flagella, yes or no? Okay, you are guessing, I believe. Yeah, these are the sperm cells, the first image. There are lots of sperms in this figure. These are the cells which have the flagella. The flagella containing cells are these sperm cells. So, on the lower side there is the paramecium, and then there is a cell which has 3 flagella. We'll talk about that later. As basically that's a bryophyte antherozoid. That's fine. Antherozoids of lower plants, they contain flagella. Sperms of animal cells contain flagella, and plant cells do not have any cell which contains flagella as such, but lower plants have that. And paramecium, it's a protozoan. It has a lot of cilia. The cilia are visible on the outside. All right. So, yeah, I can show you one more. 1 second. Okay, so alright, so this was done, and one more thing is remaining regarding the cell. Okay. All right. So this is about cilia flagella. Yeah. Now see this. Okay. Can you identify the name of this organism? Can you identify the name of this organism which is trying to move with the help of cilia and flagella? Can you identify both these cells? What are the cells that I am showing you here? If you observe that image, you can easily see what's the answer. So first cell, what is the name of the first cell? Please observe the shape. It's an actual microscopic footage of this cell. I believe the answer should be very easy and you should guess it. It's paramecium. The first cell is paramecium. And you can see the hollow spaces there. That is a nucleus. It has two nucleus, micronucleus, macronucleus. Then there is a contractile vacuole. All the cell organelles are very clearly visible in this image. But the image is not very good quality itself. But yeah, it's really good to watch under the actual microscope. Okay. So after that, yeah, the second cell is the cell called sperm cell. Okay. The second cell is the cell called the sperm cell. All right. Yeah. I hope you had guessed it, but it's okay. This is the second. This is the paramecium. This is the sperm cell. So you can see the movement that is occurring with that, with the help of that. So cilia are moving in this paramecium. You can see the image clearly. That the cilia are moving in like this. Many cilia are performing a coordinated movement, and it is pushing it in this direction. On the other hand, flagella has this kind of movement. It is having a whiplash kind of movement, like you are having something like okay. All right. Maybe if you have your wire like this, okay. So you are performing a movement like that. Okay. So this is the movement of the flagella. It's like moving. You have a whip in your hand and you're moving it like this. So flagella is having this kind of movement. It is called whiplash movement. And cilia have only one type of movement. It is going to move. Then it is going to curl inwards, coil inwards, then extend, then move in this direction, coil, and this move. Okay. So this is the movement that is performed by these structures. Okay. All right. So, and this is how the cilia help in the movement of dirt particles or the mucus particles away from the lungs. Okay. So this is an animation which will help you in understanding. So this is the mucus gland which is producing some mucus, the green colored mucus. And if you are healthy, then your mucus would be green or slightly yellowish in color, right? So everybody can take a look whenever you go to the bathroom. You can take a look at what your mucus has the color like, right? So if it is not of that color, it could indicate that you have a slight infection. So some people have white color, that is also normal. But in winters, it might turn slightly greenish or slightly yellowish in color. So this mucus is performing, the mucus gland is performing the function of producing the mucus, and just beside in the neighboring area of the mucus cells are the ciliated columnar epithelial cells, which have cilia on their surface. And cilia will catch the dirt by itself, because dirt will stick to the stick to the mucus, and the mucus will be propelled upwards through the mouth or the nose. So you know that sometimes when you're sitting in my class, suppose, and you have this urge of taking out some things from your nose, but you silently eat it and push it into your digestive system. That is also possible, okay? So normally it should be expelled from the nose or from the mouth, but when we are having this trouble, we are sitting in front of other people, we cannot do it every time. So we silently take a breath and swallow it, so that it, instead of going into a respiratory system, it goes into our digestive system. So everybody must have done it at least once in your life, or maybe you're doing it right now, okay? So that's the theory behind the movement of cilia, okay? So vacuoles I had also done. Vacuoles are large structures. They're just they are just for storing so many things. They store cell sap. Chloroplast I had also done. So this is how chloroplast actually looks like under the microscope. Okay. I had shown you these diagrams also. Okay. All right. So now the organelle that we have to do, it is the nucleus. So please put the heading now, nucleus. All right. So yeah, the control organelle or the nucleus. Okay. Yeah. So the control organelle or the nucleus, it has this kind of structure, right? So on the upper hand, the upper image, which has green color and red color and yellow color, that is the actual image seen under the microscope. Okay. So if there are some Dragon Ball Z fans here, they might recognize it as a sign of yin and yang by the Chinese philosophy, the lightness and the darkness. Okay. So it's it's looking something like that. Maybe you would agree if you know what the symbol of yin and yang looks like. Some people who are obsessed with martial arts, they might also know that symbol. Anyway, my point is, you can see that there are some things here. Something is very light green colored, and there is something that is very dark green colored. This is called heterochromatin and euchromatin. I'll discuss it, why it is having this kind of color. So first of all, the nucleus has a membrane, a double membrane system surrounding it. It is called the nuclear envelope. Nucleus contains the DNA. Sometimes their DNA appears in the form of chromosomes, sometimes their DNA appears in the form of chromatin material. Okay. Alright. It is the largest organelle of the cell. Nucleus is the largest organelle of the cell. Okay. So, more on the nucleus. Now, each cell has a fixed number of chromosomes that carry genes, but these chromosomes are not visible in the nucleus. These chromosomes will become visible in the nucleus when the cell undergoes cell division. Okay. Alright. So this is how the nuclear envelope looks like. Nuclear envelope, it has a lot of pore-like structures, lot of small hole-like structures. These are called the nuclear pores. So you can see there is outer membrane, inner membrane, and between that there is a blue colored space. It is called the perinuclear space or the periplasmic space. Normally we refer it as a perinuclear space. Okay. Alright. So all these things are clear by looking at this diagram for everyone, right? Yeah. I'll give you the notes, don't worry. I'm just coming up to that. Okay. Inside the nucleus, normally the nucleus has chromatin material only. Okay. But when cell division will happen, this chromatin material will form separate entities called chromosomes. Chromosomes we'll also discuss today. The genetic material is found as form of chromatin in non-dividing cells in the form of nucleus. But when it is condensed and wrapped around proteins, it is called chromosome. Okay. And basically what does chromatin contain? Chromatin material contains the DNA, and this information is present in the form of alphabetical letters 4 letters are used to make all the information that is contained in our cells. These are A, T, G and C, called nitrogen bases. Okay. Nucleus has another structure called the nucleolus. Okay, and nucleolus we also discussed. Nucleolus basically contains a lot of RNA material. Nucleolus basically contains a lot of RNA material. Okay, and it disappears when the cell is dividing, and it reappears when the cell is not dividing. Nucleolus is basically concerned with the making of ribosomes because it is made up of ribosomal RNA. Alright, so let's take these notes now. Okay. Alright, so coming back, please put the heading nucleus. Yeah, one more thing. One more thing remaining. Cilia and flagella both have 9+2 arrangement of microtubules. Nine plus 2 arrangement of microtubules. Okay, and cilia and flagella arise from centriole-like structure called basal bodies. Centriole-like structure called basal bodies. Okay. All right. Now we can put the heading. First of all, put a small heading regarding a centrioles, then nucleus. Well, nucleus is basically the last cell organelle we have to do. Centrosome and centrioles we will do first. Centrosome and centriole. Centrosome is basically the collection of two centrioles. Okay. Centrosome is an organelle containing two cylindrical structures called centrioles. Okay, and centriole is basically surrounded by amorphous materials called pericentriolar materials. They are surrounded by amorphous pericentriolar materials. Amorphous materials called pericentriolar materials. Okay. Now both these centrosomes are arranged at angle of 90 degree to each other. One cylinder is like this, the other cylinder, it is arranged with respect to this cylinder at an angle of 90 degrees. Okay, so this is a particular thing that you must remember. Both these centrioles lie at an angle of 90 degree to each other or are perpendicular to each other. Now it has 1, 2, 3, 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8, total 9 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah. Nine peripheral triplets of microtubules. Okay, 9 peripheral microtubules like this. 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so please draw it quickly. Okay. One more thing that needs to be drawn here. These are 9 peripheral triplets. I'm drawing only one tubule, but all the others will be like this only. This is amorphous pericentriolar material. Just a second. All right. Okay. Yeah, this is fine now. Like this. All right. You have drawn it. All right. So this is also has a all right. Please write. It also has it also has a cartwheel structure. It also has a cartwheel structure, but has peripheral 9 peripheral triplets of microtubules. Okay, so this is the structure of centrosome and centrioles. Now please put the heading nucleus. 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 So what is the nucleus? We have already discussed it. Please write. Nucleus was first discovered by Robert Brown. Nucleus was first discovered by Robert Brown. Okay. Next point. The material present inside the nucleus was found to stain with basic dyes. Basic dyes means dyes which have basic property, pH more than 7. Basic dyes, and called chromatin by Fleming. Called as chromatin by Fleming. Chromatin by Fleming. Okay. So now let's draw the nucleus. Like this. So nucleus has a basically lot of nuclear pores and so that other things. I would also need okay. So this is basically what I'm trying to tell you is this is a material called heterochromatin like this. Random segments are colored like this. And then there is a separate structure called the nucleolus. This is the nucleolus. I am drawing two nucleolus here because nucleolus can be more than one in a single cell. For the labeling, outer nuclear membrane. Outer nuclear membrane. Inner nuclear membrane. Nucleolus. Nuclear pore. heterochromatin. The material which stains darkly, it is called heterochromatin. The material which stains lightly, it is called euchromatin. Okay. The light colored material is the white colored material, and all this dark colored material and light colored material is present in a matrix called the nucleoplasm. So do you understand what a matrix is exactly? The matrix is the liquid part or liquid part in which all the substances are present inside the cell. So I can say that euchromatin can be basically like this. This is euchromatin. And the white, okay, alright. Okay, let me give you some other example. How many people know what, alright, let's consider tomato soup, okay, or let's take rice noodle soup, okay, or basically you must be knowing a lot of soups, right? So let's consider a noodle soup. If I consider the nucleus to be equivalent to a rice noodle soup, then noodles will be equivalent to the chromatin. Noodles will be equivalent to the chromatin. And chromatin is of two types, heterochromatin, euchromatin. They can be suppose some dark colored vegetables and dark colored noodles. I am calling it heterochromatin. It is very tightly packed. Some noodles have not dissolved. Okay, so that is heterochromatin. The loose noodles that are present there, that represents the euchromatin, and the liquid substance that is present in your Maggi or your soup, whatever that is, the liquid gravy which you are drinking, that is the matrix. Okay. There are two types of noodles in the nucleus. One is the hard noodle, and that I am calling it as heterochromatin. Other is the loose noodle, I am calling it as the euchromatin. And then there is the jelly-like substance or the soup-like substance in which everything is immersed into this material, that is called the matrix, and the nuclear matrix is called the nucleoplasm. Can you understand this better now? Do you understand what I am trying to tell you now, Shillong students? What is the matrix? I hope you understand now. Yes or no? Or should I give another example? Is it clear now what nucleoplasm represents? Nucleoplasm is the liquid material present in the nucleus. In that, both euchromatin and heterochromatin are suspended. Okay, fine. So this blue colored substance, I am calling it as nucleoplasm. Nucleoplasm. Right? Just a second. Okay. Now it must be clearly visible to all of you. And there is another thing I am labeling here. I am calling it as perinuclear space. The space present between the two membranes, it is called perinuclear space. Okay. Everybody has drawn the diagram now, I believe. Okay, so I'll move further. So please write, when the nucleus is in interphase stage, when the nucleus is an interphase stage when the nucleus is in interphase and it is not dividing, the nucleus consists of elaborate network of the noodle-like particles that I'm referring to, the noodle-like things, that is called the chromatin, and chromatin is made up of DNA plus protein. So I'm calling it elaborate network of nucleoprotein fibers, histone protein plus DNA, histone plus DNA, called chromatin. Okay, and chromatin material is also of two types, heterochromatin and euchromatin. Chromatin material is also of two types. Chromatin material is of two types. First is heterochromatin. Heterochromatin. And what does heterochromatin have? Heterochromatin has a dark color. It is the darkly stained. If anything is left out, please let me know. I will scroll up quickly. It is the darkly stained chromatin material. Darkly stained chromatin material. Okay. Why is it darkly stained? Because it is consisting of packed DNA. It consists of packed DNA. DNA is packed with the help of histones. Packed DNA or transcriptionally inactive DNA. Inactive DNA. Transcriptionally inactive DNA. So this DNA is not making any RNA. And if it is not making any RNA, so this DNA is not taking part in making any kind of protein. Okay? Transcriptionally inactive DNA. The other type of chromatin material, it is called euchromatin or the true chromatin. True chromatin. You can see here, this is the lightly stained chromatin material. Lightly stained chromatin material. It is transcriptionally active DNA. It is transcriptionally active DNA. Okay, it is a transcriptionally active DNA. The nucleus has a matrix called nucleoplasm. The nuclear matrix, now the chromatin material is completed. The nuclear matrix is called nucleoplasm. In which chromatin material and nucleolus is suspended. Okay. So what is the nucleolus now? What is the nucleolus? Nucleolus is a spherical non-membrane bound structure. Nucleolus, non-membrane bound spherical structure. Non-membrane bound spherical structure associated with RNA and proteins. It is said to play a role in, it is important for formation of ribosomes. Okay. Now let's come to the part called chromosomes. So now we will write chromosomes. One more thing, please write nucleolus disappears at time of cell division. Okay. Next thing is the chromosomes. chromosomes. So what is a chromosome now? If you see carefully, chromosome basically has this kind of appearance. Okay. One more thing. This is the structure of chromosome. Okay. These two are called sister chromatids. Sister chromatids. Chromosome is formed as a result of the condensation of all the chromatin material into a definite number of chromosomes. Each species has a fixed number of chromosomes that is going to be formed by the amount of chromatin material that is present there. This region, central region, is called primary constriction or centromere. Primary constriction or centromere. It's a region. These button-shaped structures here, it is called kinetochore. It is called kinetochore. This is the structure of a chromosome. Now please write. Chromosomes are thread-like structures formed by condensation of chromatin material thread-like structures formed by condensation of chromatin material at time of cell division. This is the specific time. At time of cell division. Okay. Now let's continue. Now there are different types of chromosomes based on the position of centromere. Centromere is the central region which is constricted. Okay, the chromosome has a constricted region in the center. This is called the centromere. Now centromere cannot always be in the center. Sometimes it's upward, sometimes it's a bit terminal. So based on the position and location of centromere, chromosomes are of 4 types. Based upon the position of central region called position of central region called centromere. Chromosomes are of 4 types. Are of 4 types. The first type of chromosome is called metacentric chromosome. Metacentric chromosome. The metacentric chromosome is the most easy to observe kind of chromosome in which the centromere is located in the middle. Centromere is located centrally. Centrally. Second is the submetacentric chromosome. Submetacentric chromosome. Submetacentric chromosome is a type of chromosome in which the centromere is shifted somewhere to the upper part, like this. So the lower arms are longer and the shorter arms, you can clearly see, are somewhat smaller than the longer arms. Centromere shifts towards one end, slightly, not fully, slightly. Submetacentric chromosome. Third is the acrocentric chromosome. Acrocentric chromosome. Acrocentric chromosome is a type of chromosome in which the shape of chromosome is very clearly visible. The centromere is located very close to the terminal position, but it's not terminal as of now. Okay, I should draw a better diagram for this now. Wait a second. Yeah, this is more like it. This is the acrocentric chromosome. The centromere is located very close to the one end, is located very close to one end of chromosome. One end of the chromosome. Okay. Fourth, telocentric, last chromosome. The centromere is located terminally. Is located terminally. at the last possible point. So this is how it looks like. Okay, but you can see there is small end remaining here. It is called telomere. Telomere is an extra segment of DNA that is present at one end to protect the chromosome during DNA replication, because some amount of DNA is always lost during making a new chromosome. So we always protect it by giving a safety cap. Okay. Then the last thing in this chapter, secondary constriction or satellite body. Normally, a chromosome has only one constriction called the primary constriction. But in some chromosomes there is another constriction, and after that a small body is present, and this is called secondary constriction. This is primary constriction, centromere. Secondary constriction is not given any name, but this structure present above secondary constriction, it is called satellite body. Satellite body associated with nucleus. Satellite body associated with the nucleus. So with this, our chapter comes to an end. Now let me show you some chromosomes in action. This is a normal nucleus. This is a normal nucleus. Okay, cells having the normal nucleus. But when the cells are undergoing division, they will form separate structures called the chromosomes. Okay, so just a second. A cell does not have the chromosomes at every point of time. Cell has chromatin. Chromatin material is condensed into chromosomes, and so if I take a chromosome, it will look something like this. Okay, everybody can see this. So you can see chromosome. As I am uncoiling the chromosome, I can make some chromatin, and if I am opening the chromatin also, then I will make the DNA. I will reach up to the DNA part. So basically, it has something like structure of beads on a string, and this is called a nucleosome also. So chromatin material has this kind of structure where chromosome, the DNA, double helix DNA is tightly bound upon some particles called the histone particles. So suppose this is a histone particle, I will wrap some DNA around it, then move on to the new one, a new histone particle. Okay, then I wind some things on this also, Okay, so there are two histones suppose there. Okay, so I am binding about two turns of DNA on each of the strands, on each of the histone proteins. This is the nucleosome model of the DNA. Okay, so basically this happens during cell division. Okay, and chromosome consists of two sister chromatids. If I separate them together, it becomes a single chromatid. Okay, and chromatid is a copy of the genetic material. If I separate one chromatid, it is a fully functional copy of DNA. It does not have any loss of DNA. These are the chromosomes present in our human cells. Okay, these are 46 chromosomes, and 46 chromosomes are shown as 23 pairs in this picture. Okay, 23 pairs in this picture, they are shown here. Okay, right? All right. So during the interphase stage, the cell grows in size and the DNA replicates. And this I'll explain later also how this is happening. Okay, so now let's see some videos of the chromosomes in action. Just a second. Okay. So these are some chromosomes that you can see in this picture here. The cells are dividing and the chromosomes are arranging themselves. Okay, and they are basically undergoing division of all sorts. Let me give you one more away. Just a second. Mitosis, I had, yeah. This is better. The green colored structure is called the spindle apparatus. The red colored structures are the chromosomes, and the light red color structure is the nucleus. Okay, and the spindle apparatus fibers, as they are separating, the chromosomes are separating. So please see this carefully here. That happened very fast. You can see the nucleus, it is quickly arranging in the form of chromosome. All the nuclear material quickly formed chromosomes, it happening very fast. Okay. Normally it takes some time also, but we are showing, because this process is very long, I am showing it as a quick fast forward video. Okay. And some cells which are fast dividing, they will show this kind of behavior only. You can see that the chromosomes are very quickly formed and they are very quickly separated. So in some fast dividing cells, this phenomenon will happen. And these are actually basically the human kidney cells which are dividing here. Okay. So this was about the chromosomes, and I hope you liked this. Okay, just a second. There is one more video I saved for you. Okay, yeah, now you can see this. This is mitosis happening. This is mitosis happening, and this is basically the human embryo. This is how the human embryo is formed. You can see it's a very beautiful video. Life starts from a single cell or a group of cells, 16 cells called blastula, and this is quickly undergoing division here. Here the cell growth is not happening. All the cells are becoming smaller and smaller. Okay. Cell growth is not happening actually, but basically the cells are just dividing. You can quickly see how these cells are becoming so much large in number. So this was a time-lapse video for that. And there is another cell division you must already know about meiosis. Yeah, so you can see some things here. The spindle apparatus fibers, the blue colored part is representing the chromosomes. Okay, and haploid number of chromosomes are 4. Actually 8 chromosomes in here total. So I'll explain this further also, but that needs a lot of explanation when I teach you cell division. Okay, so in the next class we will be covering this. Okay, the next class we will be covering this. So let's enjoy the video for now. Okay, and I have another video to show you. This video is basically concerning some very microscopic objects, because we have just finished the cell. So I wanted to show you some electron microscopic images first. Okay, so right. So let's see these images and have an idea of how small things can get. This is the mouth of an ant. Okay, I wanted you people to see these things, and you will find some things very interesting. So yeah, basically all this is right now boring stuff. So the real stuff is about to begin. Yeah, this is the structure of the human tongue taste receptors, taste buds. Okay, and this is a single toothbrush bristles, and the blue colored material is basically the bacteria, the plaque that is found in your dental materials. These are diatoms and dinoflagellates, and there is a sealant trait here also. It's called Daphnia. On the left side, there is an insect-like structure. It's basically a sealant trait. So water flea insects, arthropods, all these are present in this image, and this is a drop of seawater. Okay. So diatoms are present here also. This is an early human embryo, 16-cell stage, on the tip of a needle. This is a tip of a needle, a pointed structure, and this is an early human embryo. And this image is taken with the help of electron microscope. And yeah, this is a very interesting example for people who shave using electric razors and people who shave using the razor blades. So if you cut your hair with the razor blade, you will get your hair like on the left side. Okay, and if you are using electric razors, your hair will have a surface like this. Electric shavers produce this kind of structure on the hair. So you can see the smoothness is really visible in the case of hand razor, right? The electric razor shaver is having a different kind of structure. These are butterfly scales present on that. All these are present at the cellular level. So basically what I'm trying to tell you are the cells, show you the different kind of cells here. And these are the cancer cells. Okay, these are the cancer cells. These are chalk particles. Chalk particles. Okay, these are very beautiful particles. You can see that. A very small chick embryo. Okay, maybe it's looking more like an alien. Cigarette paper. Okay, and these are cut hair, like hair cut with the help of any scissors or something. Cut hair particles or shaved hair particles. They have this kind of structure. So basically these are epithelial outgrowths, right? Yeah, this is another image. Yeah, these are dust particles that are present all around you. Dust particles. Okay. So all these are things at the cellular level. This is a dust mite, and this is present all over your body on your face. This could be present right on your face, but you cannot see it because all this is look. These are very small things. Dust mite is so small you cannot see it. It is maybe present in your face right now and you cannot see it. It is so small. Okay. Dust mites. Sorry, this image has a bit of error. It's an electron microscopic image of the surface of a gold surface. This is a flea. And this is a flower pollen grain. All these are small cellular structures. So beautiful things that, yeah, this is not interesting, I understand. Human bone. You can see, you must have studied by now that human bone has a lot of canal-like structures, right? Haversian canals, Volkmann's canals. So these are visible in this microscopic image. Retina of a human. And what are visible here are rods and cones. Rod and cone cells. Okay? Alright. This is a fertilization in vitro. In vitro means this is the formation of a test tube baby. This is an egg cell, and this egg cell is being attacked from all sides by blue colored sperm cells. only one sperm cell has become successful. So now it has formed a brown colored wall around it and no other sperm can enter this embryo now. Okay. And this is marijuana, cannabis. Okay. This is basically marijuana. This is a small fracture in the steel. It's not a desert. It's not something. All these are things are observed with the help of electron microscopes. Minerals observed under microscope. Plankton. Plankton, I mean small phytoplankton, basically dinoflagellates and diatoms. Okay. This is a pollen grain. Basically, this is a wheat pollen grain because wheat pollen grain has only single aperture. Printed paper. Yeah, paper has this kind of appearance on the electron microscope. Very smooth paper, even that looks rough. And this is a bacteriophage virus. I have already told you that a bacteriophage virus, it has the capability to infect bacteria and destroy them. So this is a very beautiful image of a actual bacteriophage virus attacking a bacteria. On the surface on which this virus is sitting, it is the bacterial cell membrane surface. Okay. So you can see the head, collar and the tail fibers. Gramophone record, very old structure. And this is a very beautiful image. You can see this here. Yeah, it looks really, it's a very lovely picture. This is a ruptured capillary. Suppose you're slapping somebody. Suppose you are slapping somebody and his cheek goes red. That is because of breaking of lot of capillaries. So suppose if you're slapping somebody, lot of capillaries in his blood will break. And these capillaries, broken capillaries, blood cells are coming out. So you can see how flexible, how beautiful these blood cells are. And these are coming out from the ruptured surface of the capillary. But soon this will heal. It will heal very quickly. Okay. So you can slap your friend right now and you can see the effect, how red his cheek becomes. Okay. Refined sugar crystal and raw sugar crystal, basically gud and chini. It's a plastic fiber which can self-assemble itself. This is a snow crystal. So how many people have seen Frozen movie? Yeah, raise your hands, all those people who have seen the movie Frozen. You have seen the movie Frozen, maybe especially the girls must have seen that movie Frozen. In that Frozen movie, there are a lot of crystals having this particular type of image. That crystal line image, that is basically the representation of snow crystals. So this is an actual snow crystal. Snow crystals are formed in this way. These are very beautiful crystals. So you might remember that by the Frozen movie. Antenna surface of a beetle. And this is a tardigrade. This organism that you're seeing here, if you take it out in space, take it out on the surface of Mars, Venus, it will still be able to survive. You can take it to Mars and you do not need to give it a space suit. Take it just like that it is. It will still be able to survive in outer space without oxygen, without nutrients, anything, without water. In vacuum also, space vacuum also, it can survive. So when it has lost all water, it is going into dormant state and it can survive. Textile fat materials, fabrics like clothes that you're wearing. Okay. This is a squid, and squid has a lot of suckers by which it's it's a maggot. Maggots are formed from flies, house flies and so many insects. This is a shark skin. Sharks have a lot of scales, you remember? Sharks have placoid scales. Bony fishes have ctenoid and cycloid and ganoid scales. So this is sharks, and it has placoid scales, triangular placoid scales. Tardigrade, normal healthy one without losing water. It also looks like a small monster. It is very small. Tooth surface. I don't think somebody has a tooth that are this dirty. Alright. So these are some beautiful images I wanted to show you because cell chapter is finished now. And after this chapter, we will do a lot of things. This is Velcro fiber. The Velcro that sticky material that we use. Wood fibers. Yeah, this is a very beautiful image I wanted to show you. What do you identify in this image? Can you identify what is present in this image? These are sclerenchyma fibers and xylem vessels. These are xylem vessels and sclerenchyma fibers, okay? Right. So, it's nearly over now. Fabrics only. Alright, so thank you for watching this. These were some cellular images I wanted to show you. I hope you enjoyed it. So I'll see you in the next class and then we will be starting cell division. I'll give you a PPT for that also and I'll send you these notes on your WhatsApp group. Okay. Thank you dear students. We will meet in the next class. Thank you.